You may be 60 years old, but that does not mean every part of your body is 60 in the same biological sense. Your lungs may be aging faster than your heart. Your pancreas may look older than your brain. Your muscles may remain relatively well preserved while your blood vessels show structural changes normally seen much later in life. A major Nature Medicine study published on August 14, 2026, has now made these differences visible under the microscope. Researchers used artificial intelligence to analyze more than 25,000 tissue images from 40 different human tissue types and trained models to recognize what aging physically looks like inside the body. This is different from most biological age tests. Many aging clocks examine DNA methylation, blood chemistry, proteins, or medical imaging, and then compress that information into one number. This study looked directly at tissue architecture. The AI learned from microscopic pathology slides containing the actual structures of organs, including blood vessels, connective tissue, muscle fibers, glands, epithelial cells, and surrounding support tissue. Instead of asking only how old a person was, researchers could ask a more interesting question. Does this particular tissue look younger or older than it should for that person's chronological age? That distinction matters because aging is not uniform. Imagine two people who are both 60. One person's major organs could mostly resemble tissues expected in the mid-50s. Another person's heart, lungs, pancreas, and arteries could look several years older than expected. Their calendars say they are the same age, but their bodies may have accumulated very different amounts of structural damage. Researchers increasingly find this kind of organ-specific aging using blood proteins, MRI scans, and other methods. The new study adds something different: visible microscopic evidence that separate organs can follow different aging trajectories. The team analyzed 25,712 whole slide histopathology images covering 40 tissue types from 983 people. The AI models were trained to estimate chronological age from the appearance of those tissues. Once the system learned the normal relationship between tissue structure and age, researchers could calculate an age gap. If tissue from a 55-year-old looked more like tissue normally found in a 65-year-old, that organ had a positive biological age gap. If it looked more like tissue from a younger person, the gap moved in the opposite direction. Across tissues, the models were able to estimate age with an average error of roughly 5 years. But predicting birthdays was not the real discovery. The researchers wanted to know what the AI was seeing. When they examined the microscopic structures associated with older-looking tissue, recognizable signs of biological deterioration appeared. Aging tissue showed patterns involving atrophy, increasing fibrosis, loss of small blood vessels, deterioration of epithelial structures, and changes in tissue composition. Skeletal muscle, for example, increasingly showed fat infiltration. In the aorta, older tissue showed structural changes consistent with vascular deterioration. In the brain, aging signatures included changes associated with tissue integrity and blood supply. The clock was not simply finding some invisible mathematical pattern. In many cases, it was detecting physical deterioration that pathologists already recognize as biologically meaningful. This leads to the first important twist. Biological age may not be one property of the entire body. It may be a collection of separate but interacting ages. Your lungs experience air pollution, cigarette smoke, respiratory infections, and oxygen exposure. Your liver processes nutrients, medications, alcohol, and metabolic waste. Your arteries experience mechanical pressure every second of your life. Your muscles respond to whether you use them or not. Your pancreas is repeatedly challenged by the need to control blood glucose. There is no reason these tissues should accumulate damage at exactly the same rate. When researchers examined several tissues from the same individuals, they found different aging patterns. Some people appeared relatively resilient across much of the body. Their tissues tended to look younger than expected. Other people showed widespread accelerated aging. A third group showed something more selective. One or several tissues looked unusually old, while much of the rest of the body remained closer to normal. This is important because a single whole body biological age number could hide exactly the organ that is beginning to fail. Imagine receiving a report saying your biological age is 58 when you are chronologically 60. That sounds reassuring. But suppose your brain appears 54, your muscles 55, your liver 58, and your pancreas 69. The average might still look good while one organ is moving in the wrong direction. The emerging science of organ-specific aging suggests that future health monitoring may look less like a single age score and more like a dashboard. The next question is whether these tissue ages actually relate to disease. The answer appears to be yes. The study found associations between accelerated tissue aging and several chronic conditions. Chronic respiratory disease was linked with older-looking lung tissue. Type 2 diabetes was associated with accelerated pancreatic aging. Cardiovascular and metabolic disease showed patterns involving relevant tissues. Some systemic diseases were associated with aging in organs beyond the primary site of illness, reinforcing the idea that chronic disease can affect the body as a connected network rather than remaining isolated inside one organ. That network effect has appeared in other aging research too. Plasma protein studies have shown that people can have unusually old or unusually young individual organs, and the number of accelerated organs is associated with future disease and mortality. One major study found that an especially aged brain was strongly associated with Alzheimer's disease risk, while people with youthful brain and immune system profiles had lower mortality and were more likely to reach exceptional ages. These methods are completely different from analyzing tissue slides, yet they point toward the same conclusion. Organ age contains information that chronological age misses. Then the researchers attempted something even more ambitious. Tissue biopsies are invasive, and nobody is going to remove samples from 40 healthy organs every year just to calculate biological age. So the team used paired gene expression data from blood to see whether signals circulating through the body could help predict the tissue-specific age gaps observed under the microscope. They then tested these approaches in independent disease cohorts. The predicted aging patterns often matched the systems affected by disease. Alzheimer's disease showed strong brain aging signals. Gastrointestinal inflammatory diseases showed aging patterns in relevant digestive tissues, and vascular disease affected highly vascular organs. If that approach eventually becomes reliable enough, it could create a completely different type of health assessment. A normal blood sample might someday estimate whether particular tissues are aging unusually quickly before severe symptoms develop. Instead of simply being told that you are biologically older than average, a clinician could potentially see which system is responsible. That information would be much more useful because different organs require different responses. But this is where the hype needs to stop. The technology is not ready to tell healthy consumers the exact age of every organ. The study has important limitations. Much of the tissue data was cross-sectional, meaning researchers compared tissues from different people at different ages rather than repeatedly sampling the same organs in the same individuals for decades. Pathology slides can also vary depending on how tissue is collected, preserved, stained, scanned, and processed. AI models can accidentally learn technical differences between hospitals or laboratories instead of true biology if these factors are not carefully controlled. Previous digital pathology research has demonstrated that site-specific imaging signatures can bias AI performance. There is also a deeper problem with all biological age clocks. Knowing that an organ looks older does not automatically tell us how to make it younger. An old-looking pancreas might reflect metabolic stress, fat accumulation, inflammation, vascular changes, or loss of functional cells. An old-looking lung could reflect smoking, pollution, chronic respiratory disease, infection history, or other factors. The number is only useful if doctors can identify the mechanism behind it and intervene safely. This is why an aging clock should never become the goal itself. If an intervention makes your biological age score fall by 5 years but does not reduce disease, preserve mobility, improve cognition, or extend healthy life, then the younger number means very little. Nature Medicine recently emphasized exactly this broader problem. Anti-aging interventions need stronger clinical evidence and reliable biomarkers that reflect meaningful health outcomes rather than marketing claims. For people today, the practical message is much less futuristic. We already know several major exposures that damage specific organs. Smoking accelerates injury to the lungs and cardiovascular system. Poor metabolic health affects the pancreas, liver, kidneys, blood vessels, and other tissues. Physical inactivity contributes to loss of muscle and cardiorespiratory fitness. High blood pressure damages arteries and organs over time. Managing these factors remains far more useful than buying an unvalidated commercial biological age test. But the study changes how we should visualize aging. We usually imagine the whole body gradually becoming older together, almost like a photograph slowly fading. The new picture is more complicated. Aging may resemble a city in which individual neighborhoods deteriorate at different speeds. One district remains well maintained, another begins losing infrastructure. A third suddenly accumulates damage after disease or years of environmental stress. The city's average age does not tell you where the next failure will happen. That is why organ-specific biological age could eventually matter. Instead of treating a healthy 60-year-old and an unhealthy 60-year-old as simply the same age, medicine could identify the systems where each person is unusually resilient and the systems where risk is accumulating. Monitoring could become more targeted. Prevention could begin earlier. Treatment could potentially focus on the organ that is actually moving fastest toward disease. The biggest discovery is therefore not that AI can guess someone's age from a microscope slide. The important finding is what happens when the AI gets the age wrong. That error can contain biological information. A tissue that looks older than its owner may already be carrying structural evidence of accelerated decline. Your birth certificate gives every organ the same age. Your biology does not. And as scientists learn to measure those differences more precisely, the future of healthy aging may become less about asking how old you are and more about finding out which part of you is getting old first.